My name is Brianna. I'm doing my PhD at Charles State University in Wagga, and I am the fortunate recipient of the Angus Foundation Research Grant. While I grew up with limited knowledge of the beef industry, I was always passionate about research, particularly in animals. This passion eventually led me to CSU, where I did my undergraduate degree in animal science. This is where I started to learn about the different livestock industries, but it wasn't until an honours project based on cattle was offered to me that I began my research career in the beef industry. So in my honours project, I focused on researching the changes of microbial communities, otherwise known as profiles, in beef cattle as they were placed on feedlots. Feedlots can lead to increased disease susceptibility for many cattle, and there are lots of different factors which can influence this. As we are starting to learn in the research field, microbes are amazing organisms that have the ability to influence a wide range of processes within the body, including digestion, metabolism, and immunity. Now, this is starting to be applied to livestock, with studies being published that show microbes can influence similar processes in cattle. My honours research showed that gut microorganisms changed on day 2 of feedlot placement, and this shift could impair the cattle's immune responses and cause them to become more susceptible to disease during this period. This project and the results discovered led to my passion for microbes and what they could do potentially for the beef industry. We now see microbes as a potential way to indicate and influence the health and productivity of beef cattle. As the research is still quite new and the door was open for further study, I began my PhD. My PhD project is currently focused on investigating the relationship between fecal microbial profiles and the production traits such as weight gain and immune responses in Angus beef cattle. From the start, my project was only possible due to Angus Australia's sire benchmarking program. This collaboration came about because the Angus herd at CSU is annually benchmarked by Angus Australia, and we saw this as a good way to combine our resources and essentially design a better PhD project. As this was successful, I was able to access CSU's Angus cattle herd, sample fecal matter during benchmarking, and get data for my project including weights and immune response phenotypes. Even with this collaboration, my project method was still weak due to funding limitations and sampling size. However, in 2021, I was awarded the Angus Foundation Research Grant, and this has allowed me to greatly strengthen my project. With the help of the grant, I was able to sample an additional 270 animals. This larger number of samples meant the immune response phenotypes could be split into 3 groups, high, medium, and low, increasing the project's statistical power. Not only could we now get a better picture of what was going on within the microbial profiles, we had a big enough sample size to create an additional objective for my PhD, to estimate the heritability of specific microorganisms. So here are some early results from my PhD. On the right are 2 plots from my weight study, displaying how many different types of microbes there are in a group, referred to as richness. It can be seen that the weight groups at weaning stage have similar species richness. However, at yearling stage, they are separated, with the high weight group having noticeably lower species richness. Previous studies have suggested that cattle with lower species richness have increased feed efficiency and average daily gain. This is exactly what we are seeing here at yearling stage. So therefore, this could be a marker for weight performance in Angus cattle. Our next step is to determine how early this can be seen. The plot on the left displays the relative abundance of a specific microbe found to be differing between the high, medium, and low groups of the antibody-mediated immune competence phenotype. I found that this microbe was significantly more abundant in the high immune group, and previous evidence has suggested that it has a protective effect in the gut and may even contribute to immune responses. Therefore, this microbe may be a marker for high immune responses in the future as well. Furthermore, we are currently determining the heritability of specific microbes in relation to each phenotype, and in these early stages, we have some microbes that are appearing to be heritable. If we can determine that microbes influencing immune responses are heritable, then this may open an alternative avenue for improving beef cattle production. I believe these results will eventually contribute to and improve the beef industry in unique ways. I love this work and would love to continue contributing to the beef industry in this way into the future.
So what does investing in people mean to you? How has that been for your journey. You're now working in the beef industry. What happened?
Yeah, no, definitely. I came from no agricultural background at all. My family doesn't work in the agricultural business. I just had a passion for animals. And so if I hadn't been offered an opportunity within the beef industry, like I wouldn't be here. And I think I'm lucky that I am here now because there is a big avenue for growth in kind of what I'm doing within the beef industry. It's been done a lot in humans, but we're not quite there yet with animals. So I'm really keen to kind of take this forward and see what it can do. So I definitely wouldn't be here.
Brianna, I know one of the things you're passionate about is um, the constant battle for research funding. But if we're talking about producer or industry through the research and battling for funding or direction, or how have you seen that challenge, I suppose, in your early career?
Well, I mean, as you probably just saw, my project wouldn't have been what it was if I hadn't had funding. And I think now we can really explore a lot, I guess, more relevant things to the beef industry. I know research usually starts in a little bit of a weird corner, and a lot of producers may think that it's not quite what you need on farm, but I think we're coming up with unique, I guess, solutions, and they sometimes can't be brought across in the correct ways. So we kind of start somewhere, and then we can't get that out, and we also can't get any feedback back about what producers want. So yeah, I reckon crosstalk there does need to get better, but also if we have more funding, we can kind of do more things and get to the goal a little bit quicker, and that's what everyone wants. We just want to, make beef production better, I guess.